I'll do my best. Okay, so let's talk about therapeutic cloning, also known as stem cell treatments or regenerative medicine. Okay. So this is where we clone specific tissues or cells. Okay, and then we use them as a therapeutic, which just means as a treatment for some disease. Okay? So we're going to talk about stem cells. Stem cells are cells that can become other types of cells, right? In a human being, in an adult, right? Most of our cells are adult tissues. And we're not doing a lot of cell division. We're not making a lot of new cells, except in our bone marrow, right? Remember I said we're making blood cells every day, red blood cells, white blood cells. So bone marrow cells are considered stem cells. They're adult stem cells, right, because you're no longer an embryo. So we don't call them embryonic stem cells because they're in an adult, right? But they're definitely a tissue where they start as a bone marrow progenitor or a blood cell progenitor, something that comes first, and then they sort of grow up in the bone marrow to differentiate or become red blood cells. So the same cell in your bone marrow can become a red blood cell, can become the 20 different white blood cells, can become a platelet, right? From one cell. That one cell that can become all those other blood cell types is called a stem cell. Okay? That's an adult stem cell because it's in adults, and we are making those new cells every day. Okay? What normally we think about stem cells are the embryonic stem cells, cells that can become every other cell type in our body. They have to. We all started as one fertilized egg, one cell, right? Egg, sperm, woo, gets together, 23 plus 23 equals 46. That one single cell became you, your 20 gazillion billion different individual cells. Every single cell type in your body exists from one original cell. Okay? So that is the epitome of a stem cell. It can become everything, and it did become everything, because you're here, right? Okay. So that's an embryonic stem cell, right? The original fertilized egg can grow into an entire human being. Okay? They can make every single cell type. Okay. Yes, sir? So his question is, so hypothetically, if we took some stem cells and put it into somebody's leg, could they regrow their leg? It's within the realm of possibility if those cells get all the right signals that normally happen in development to make that happen. Okay, and so we'll keep talking about that and how we can regenerate tissues and what ones we can do right now and what tissues we're trying to work towards, right? Anything complex that has lots of different types of tissues, like a leg, right? If you think about a leg, it's got to have all the veins, it's got to have muscles, it's got to have all the nerves, it's got to grow a bone, and it's got to be in all exactly the right shape. It's unlikely that we'll be able to just stick stem cells in, an amputee's and have it grow a whole new leg, but it will be possible to grow the muscles, the vessels, the nerves, and all the stuff you might need, and then potentially put them around some sort of structure, right? Instead of growing the bone, you would use some titanium setup and build it around that mesh of structure, right? And so we'll talk about all the things we can do right now and then all the things that we're going to try to do. But first, let's talk a little bit about the process that will help you understand a little bit better of why some things seem impossible and why some things are definitely possible and are happening now, okay? And so what normally happens is the embryonic stem cells we get, we don't take the fertilized egg, okay, because that's one cell. We let that grow up to a stage of about a ball of 8 cells or so, okay? And because it's much more stable. So These embryonic stem cells that are used for treatment and research come from in vitro fertilization attempts. Okay? And so let's first talk about in vitro fertilization and then talk about how that gets to a research lab or to the clinic. So in vitro fertilization is just something that couples can do if they can't have a baby the good old-fashioned way. Okay? There's some problem with ovulation, or there's a problem with the sperm swimming, or there's some other issue that's going on, and couples are infertile, they cannot have a baby, right? We can take the eggs from a female and the sperm from a male and do the fertilization event in a petri dish. So in vitro just means like in a test tube, outside of the body. Okay? So we can, instead of it happening up in the fallopian tubes, right, we can do this in a petri dish. Okay? And so the process would be in the females, they take a whole shitload of fertility drugs so that they super ovulate a whole bunch of eggs, and then those eggs are surgically removed, right, and put into the petri dish. The sperm is much more easily acquired, just a magazine maybe and a closed door. Okay, that goes in here. It's in nutrient media, right? The sperm do their job, get in there, fertilize those eggs, and then these are let to replicate until they get to a decent size. It looks like they're going to continue development, and then they're reimplanted into the female. Okay? Usually, in vitro fertilization, now they're getting really good at making it work. No more than 3 embryos are put in at any given time, right? Because the human beings are supposed to have 1 baby, right? Sometimes twins. The occurrence of a natural triplet that's not been messed with any drugs or something is extraordinarily low, but it is possible for a human being to have 3 in just by good luck or bad luck, however you might want to look at it. Okay? But extraordinarily rare. Any more than that is not acceptable for people, right? We're not supposed to have litters, right? Animals that have a litter have uterine horns, which are very long, and they're set up to have 6 on each side, right? We're set up to have 1 baby in there, 2 at the most. 3 becomes usually dangerous for the mother and the offspring, okay? So they try to not put more than 3. If the person's relatively young and healthy, they'll only do 2 in the first round. So lots of in vitro fertilization results in twins, okay? So if they put in 2 and they both stick, you get twins. If they put in 1 and it splits, then you have identical twins, okay? And in vitro fertilization, the event of identical twins is much higher than in the good old-fashioned way, right? So the identical twin population has risen due to this, right? So if they put in 3 and they all stick and 1 splits, then you have 4. And that could be, or more than 1 splits. And then that becomes dangerous to the mom and to the babies. So we're going to watch a little video clip. Do you think it's educational? Oh, you guys are brilliant. Okay. So, let me say one more thing. So if the problem is something with the sperm, right? So in a lot of cases, the egg was fine, but there's a problem with the sperm. Instead of just dumping the tube all over the eggs in here, they can actually inject a single sperm into an egg. So if the problem is the sperm can't get in the egg, like that's its malfunction, then they can actually poke it in there and help it out. Okay, so the little video, I think, is going to show us that single sperm injection, which, as it turns out, is a lot more expensive than when they just dump it all over the eggs in there. But it works quite well.
In preparation for in vitro fertilization, a human oocyte is held in place. In this technique, intracytoplasmic sperm injection, a single sperm is drawn into an injection pipette. Gentle pressure is applied to break through the tough outer cell layer and insert the pipette into the cytoplasm. Once inside, the single sperm is released and the pipette is carefully withdrawn.
Pretty awesome. So then if that goes through, right, the proper development, it will become that ball of cells, and then we can implant that into the uterus, right? If we know anything about reproduction, right, the egg and the sperm normally meet in the fallopian tubes. It takes about 7 days for it to travel down to the uterus to implant. Okay, and so in that period of time, it's replicating. So the implantation event is usually between, 8 cells and 16 cells when it actually implants. So it's about a week after fertilization. Okay, and so they try to mimic that in the same way doing in vitro. Any fertilized embryos, the ball of cells that look like they're functioning healthy, that are not being used, are frozen. So they can be frozen same with in that minus 300 and whatever Fahrenheit liquid nitrogen tank, just like the zoo, they said, right? So the same kind of cold, those embryos can be frozen and saved for later. So let's say they do the implantation, couple has a baby or two, 3 years later, they're crazy enough to do it again. They go back into the freezer, they pull out 3 more, they do that. At some point, after they've had enough children, they're done. Okay. At that point, those embryos that are left over are in the freezer. And at that point, the parents are given the choice, what do you want to do with these embryos? Do you want to keep them frozen forever, which costs you $1,000 a year or something? Do you want to donate them to another couple that's infertile, that their in vitro didn't work for whatever reason, the egg and sperm were so messed up they couldn't make it happen, right? Or do you want to donate those to research? Or do you want to throw them in the trash? Okay, so those are the choices. So it's up to the parents whether these fertilized embryos go to research or not. Okay, so they have to be informed consent and all sign a million papers and so forth. Okay, so that's the choices when somebody has leftover IVF. So that's where the embryos come from to do any kind of embryonic stem cell work. Yeah?
Now, when it comes to abortion, when is this considered abortion? At what stage?
So her question is about abortion, and what is this considered, would it be considered abortion in like a real person?
Yes.
So at the point of fertilization and implantation, right, you have no idea you're pregnant, unless you're really, in tune. Your pregnancy test does not become positive until a week after implantation. Okay, so no. So that at the stage, once they're good enough or more developed enough to have a good implantation, you can no longer get embryonic stem cells from them. Okay, so they've gone through that stage where you could get embryonic stem cells. So if somebody peed on a stick, has a positive pregnancy test, and then runs and says, I want to donate this to research, it doesn't work. It's already too late. Okay, so you can only get those cells before anybody knows they're pregnant. And they're never taken from a real person or from any aborted fetal tissue. They can use fetal tissue to do therapeutics, right? Like or miscarried, however you want to say it. If you donate that tissue, right, just like being an organ donor, if you had, a miscarriage and you were at the hospital, you could donate those tissues to be for organ donors. So usually they're used for cornea transplants, right, eyes, occasionally for liver, anything that you don't need an entire organ for, right? Because the fetus is way too small to do a liver transplant or something like that, but you can get tissues from them to do some therapeutics. But again, that's by parental consent if they want to donate that or to research to study those types of transplantations. So yeah, so to get embryonic stem cells, it cannot come from any person. It has to come from an in vitro fertilization. Okay, and right now, you can't do in vitro fertilization just to get embryonic stem cells to do research. They have to come from a donated IVF lab. So just because you're in a lab, you can't say, okay, I want to donate my eggs, and then my lab partner is going to donate the sperm, and we're going to make some embryos, and then we're just going to study them. You can't do that either. So they have to come from donated individuals. And for the most part, that's relatively rare now, because what we've been able to do is take those embryonic stem cells from those few embryos that they get and create what you call a cell line, grow them in culture essentially forever, right? Because they're stem cells. Their thing is to make more of themselves. And if you grow them in culture under the right conditions where you're not giving them any developmental signals, they will stay as embryonic stem cells. They won't differentiate into any of the other tissues, right? So you can grow up 1 billion of them, take a couple 100 million, do your experiment, and keep growing those cells so that you don't go back to a fertility clinic to get more embryos all the time. Very, very few labs actually get live embryos now to extract the cells from, right? There's a couple labs in the whole country that does that, and they provide the cells to every other researcher in cell lines. Okay, so it's very rare to actually use a real embryo anymore. So most of them are these cell lines that they're using. And then those you can freeze down just like any other types of cells. So we use cancer cells in labs to study things as well. Those can be frozen, so you can keep them forever, bring them back out, grow them up, and that sort of thing. So you can perform all the studies without ever destroying any embryos. Okay, and so then this would be the overall idea of stem cell therapy, right? So you grow, you do the in vitro fertilization that's donated. You get to the point which they call the blastocyst, and that's the thing that implants into the wall of the uterus, right? And then that little mass that's inside, that's the stem cells. That's what becomes the baby, okay? The ring, the cells around the ring, that becomes the placenta inside the uterus, okay? And so what happens is those, as soon as implantation happens, those cells start developing into the fetus, right? So right after implantation, they start changing, they start making the different layers, right, to make muscle and bone versus, you know, all your inner organs, the ectoderm, endoderm, mesoderm, all that baloney during development, so that by the time the person knows they're pregnant, that's already been developed beyond the stage where you can get those embryonic stem cells out, right? So they have to be very, very early, before, essentially at implantation or before, in order to get those, okay? And so if you take out that mass of cells, right, we can grow them up, right, make a cell line out of them and keep them growing forever. Take some of those cells, we call those pluripotent stem cells. That means they can become almost everything in the body. The only thing those cells can't become is the placenta, because those cells were already differentiated into that outer ring. So pluripotent just means become every different cell type except the placenta. Totipotent, or totally potent, means it can become every single thing, including the placenta, and that would have to occur at the fertilized egg stage, or before it makes that little ring out there. Okay? So embryonic stem cells that are used for therapeutics are pluripotent. That just means almost everything can become almost everything. Everything we need, like especially boys, right? You're not going to need a placenta, I hope. Okay? Right? So every kind of cell type that we need. We know, scientists know, I don't know, all of them, how to differentiate lots of different cell types from those embryonic cells. The exact nutrients, hormones, proteins to put into a flask to grow nerve cells, to grow heart cells, to grow more skin cells. Okay? We can grow almost every single cell type out there now, or very close to it. Okay? We can do this in mice, right? The same idea, in vitro fertilization for mouse, grow up some nerve cells, right? Take a mouse that has a spinal cord injury, inject nerve cells into the spinal cord, they engraft, they heal, the mouse walks again. We have cured paralysis in model organisms using stem cell therapy, right? That's one step away from doing that in human beings. That's power, right? We can make heart cells in mice, and we can give them to a mouse that had an induced heart attack, where there's a lot. Heart attack is just dead heart tissue, right? If too much of your heart is dead, guess what happens? Yeah, you're dead. Okay. So if you survive a heart attack, it's because not enough of your heart tissue died to actually kill you. It can still function, right? It's never going to be the same if you didn't have a heart attack, because there's a patch of cells that are dead, right? We can take in a mouse, we can take those heart cells we made in culture, jam them back into the heart tissue. The heart's actually in this little membranous sac. You just fire them in there. They engraft. They heal over that scar tissue. Mouse as good as normal before the tissue. One step away from doing it in people. Okay? And about $4 billion. Yeah. So that's a great question. So is this why sometimes they take the cord blood from the baby when you give birth? Yes. So cord blood only has the bone marrow stem cells in it, okay? Because it's actually very rich in that. So the baby's bone marrow and the bone marrow actually is in that cord as well. And so they save that, and that's like saving extra bone marrow for that child, right? And so that if that child needs a bone marrow transplant later or needs gene therapy done, something like that, then yeah, they can take that out of the freezer, manipulate it, and put it back into that child. The question is, can they use that for the parent? Let's say something happens to one of the parents. It's not as helpful because your immune system will see it as foreign because it's not yours, right? And so that's the problem with any kind of transplantation. Like if you know somebody who's had a kidney transplant, they have to take immunosuppressive drugs the rest of their life because their body will reject it as foreign, right? Your immune system job, anything that doesn't belong needs to get kicked out, including cells, and that's bacteria, viruses, to keep us safe. So we have to kind of ramp down the immune system in order to keep any kind of transplant that didn't come from you, right? So even if it's your kid, they should be about half as good as you, right? But still not sometimes not a great match, especially if your partner is totally different than you, and right? You're better for transplants as a sibling. You're actually more genetically similar to your siblings than to your offspring, right? Because you shared the same mom and dad. You didn't get the exact same half of dad and half of mom, or you'd be unless you're identical twins, right? But it's usually much closer than a parent to a child. Okay, so let's watch this, another educational video. What the hell's happened to me today? Oh no, off my net.
Dr. John Gearhart was at the forefront of that achievement.
These cells have two properties. One is that if you keep them in the dish under certain culture conditions, they will continue to form more cells like themselves. So you can grow a room full of these embryonic stem cells, and they are undifferentiated cells. They all look alike. If you take some of these cells, though, and you put them out in different kind of growth conditions, these cells are capable of forming all the cell types that are present in the human body.
What we are looking at here are heart muscle cells that are beating as a tissue.
Once, these were stem cells with uncharted destinies. Dr. Gearhart has directed their development into heart cells, now able to beat in perfect synchrony.
Yeah, that's what I'm talking about, people. Okay, so why can we do this? Why can we do this with a mouse, right? A lab mouse, and put the cells back in, and it cures their heart disease, and they're fine? Like, why don't they reject the cells? Because mouse lines are so inbred, the ones we use in labs, they're essentially clones of each other. Okay? They've been inbred for so long that their DNA is almost identical, and so between two different lab mice of that same strain, they don't reject each other's tissues because they don't see it as foreign. Okay? So the biggest problem with embryonic stem cell therapy right now is that it's not you, right? Even if it's half you, it's not you. That tissue, we grow up and make heart tissue for you, even if it was from your embryo, right? It's not going to be, your immune system is going to still see it as foreign. Okay? So the proof of principle that this can work is established well in these model systems where all the organisms are essentially clones of each other, right? So we can avoid that whole immune rejection. So we know it can work if we can just get to the point of no immune rejection, right? Somehow do that without using, somehow make the same kind of embryonic stem cells, but not from an embryo that's not you. Okay, and so the biggest problem with stem cell therapy, even though it's got this enormous potential to do all kinds of amazing stuff for almost any disease you can think of out there, is that the federal government is scared of it. Okay? It smacks of very, very science-y, and science-y stuff and big words scare politicians. Okay? And they also have problems with religious groups who are under the assumption that scientists are running around having women get abortions to steal babies to do stem cell work, which, as we all now know as educated Bio 100 students, you cannot get embryonic stem cells from any aborted tissue. Okay? You can only get it from the fertilized egg that's grown before it can implant into the uterus. Okay? And they all come from donated embryos that are left over from people who no longer need them. Okay? But the whole when does life begin and when is it murder and so forth is a huge issue, right? A huge ethical issue, and politicians are scared to generally make a stand one way or the other. Okay? So the Republicans make a stand and say, no embryonic stem cell research is allowed. We're not going to fund it by the federal government. Okay? The Democrats, generally a little more liberal, right? That's our thing. And they say they're going to allow some of it, but then usually Congress says no. So up to this point, there's been very little federal funding for any kind of stem cell research. Most medical research that happens in this country is funded by the government, the National Institutes of Health. That's who we write grants and beg them for money and try to convince them that what we're doing is good. Okay? So what happened is the states took it into their own hands, and several different states came up with their own funding sources to do stem cell research. Okay? So, no, I'm lying. So, in California, the California Institute for Regenerative Medicine was passed on the ballot in 2006, and we all voted, well, you guys were probably too young, for $3 billion in bond funds to go towards stem cell research in California. And so California, Massachusetts, I think Connecticut, and a couple other states have the same, Michigan maybe, the same types of funding so that we can progress in stem cell research. Okay, so this funding source gives out a lot of money. As it turns out, we here at CSUN have a bunch of money from them. And so through a grant that I wrote with my friends at UCLA, we have $3.4 million to do stem cell research at CSUN. And so what we do with that money is we send our students over to UCLA to use all their good stuff and all their expensive stuff, right? So we're totally like mooching off of them because they have it all set up. They have an entire building built with this CERN money, right, with all the high-tech, most expensive equipment that you could get, and so we just mooch, right? We send our students, I send them with a whole bunch of money, right? They get paid a little stipend, they get their tuition paid, and they go over there and they work their butts off, okay? And so what a lot of the students are doing, one of them was working on this mouse project with the heart cells, right? There's a couple students that work on the mouse and the paralysis. There's Parkinson's disease. There's all different metabolic disorder stuff that they're working on. And one of the biggest things that they're doing is not only are they looking at the embryonic stem cells, right, which can become anything. One of the, so here's just a little review of the different types of stem cells. So if you're a single-celled fertilized egg, that's the only thing that's totipotent, right? Can become everything, including placenta, right? If you're this 5 to 7 day old embryo with the ring around it and the inner cell mass, the ring is already going to be placenta. The inner cell mass is now pluripotent, become everything except placenta, right? If you get cord blood or adult stem cells, these are considered multipotent, right? Because those can only become more blood cells, right? They're from your bone marrow or the cord, right? They can become red blood cells, white blood cells, platelets, but they can't become nerve cells. They can't normally become heart cells or any of the other things in their normal differentiation. Oh my God. So everybody's good with totipotent versus pluripotent versus multipotent, right? So think of as multi as most, pluri is almost everything, and toti is toti, total. We also can find some stem cells in lots of different tissues, right? They're really, really difficult to isolate, so it's not like something we just stick a needle in and suck a bunch out like we can with bone marrow, right? But there are scientists trying to isolate, well, the bone marrow is easy, like heart cells that are already the stem cell in there, right, to use. Because then that way you could take out your own cell, grow them up in culture, and then give it back to you. Right? Then you wouldn't have any rejection problems, but that's really, really difficult. They're really, really hard to find. The best thing that's out there now, the best hope for therapies is something called an induced pluripotent stem cell. This is what lots of our students from CSUN are working on right now over at UCLA. This is where you can take a skin sample from somebody. So I just come over and I just punch biopsy, right? Just take a little chunk of your skin out. And you're like, what the hell? And then I stick it in culture, right? And I essentially do gene therapy on it. I give it a bunch of DNA protein factors that have it reprogram itself and it, oh my God, I think I'm a stem cell. Okay? And those cells actually change what proteins they're making, right? Their gene expression, they turn off their skin cell program and they turn back on, I'm an embryonic stem cell, I can now become anything. Then we can take those guys and drive them to become heart cells or nerve cells or skin cells or pancreas cells to treat diabetics. Okay? It's your own cells, you won't reject those because they're yours. Your immune system will see it as you, right? So if we grew up heart tissue for you because you had a heart attack, we can just jam it right in there. Well, it's not that easy, but right? And we will not have an immune rejection. It's your cells, right? This is where the therapies will come from. Okay? So we can do this in mice and all the model organisms and grow them up, and yes, it does repair nerves damage, spinal cord damage. Yes, you can have them engraft into heart tissue and help with heart disease. Yes, you can cure a diabetic mouse by doing this process. So again, this is an even closer step to doing this in human beings. There are some clinical trials now doing this for paralysis. Okay? Clinical trials are really, really expensive because you want to make sure everything's perfectly safe. They do all the tests in the flask first, and then in an animal model, and then in people, right? And the people obviously have to be volunteers, right? So they're volunteers, they know exactly the risks and the possible benefits, and they're willing to take those risks to do those types of clinical trials. Okay. The biggest problem with the iPS cells is the gene therapy part, is getting that DNA into those cells safely. Okay? And so what we found is, right, we introduce these genes, treat with small molecules, right, grow and differentiate, we get all of this stuff, we put them in animal models, and maybe 1 in 100 of those mice that we treated get cancer. Because those cells started growing out of control. Right? Cancer is just cells growing out of control. 1 in 100 mice, not a problem. 1 in 100 people, not a good number. Okay? And so that's the problem as far as getting from the lab to the clinic, is solving that problem of how can we avoid using viruses here, right? Or this process of, are all of these now really nerve cells? There's nothing that's still a stem cell that could become something else like a tumor. Right? That's where we're stuck right now in therapy to get to human treatments. Okay? And so just like the human embryonic stem cells, right, we can get these induced pluripotent stem cells from any adult tissue, turn it backwards, make them think they're embryonic stem cells, and then grow them into anything else. It's the safety issues that are being dealt with now. All of those things are really expensive. All of those things take lots of money, and the federal government is still not funding it. Yeah. His question is, what causes cells to grow out of control? It's usually some kind of mutation that happens, usually. When we're going to start talking about cancer in the next couple weeks, and we'll talk about how mutations can happen, usually in a specific type of gene or more than one that is supposed to be controlling the cell division, and it gets out of control. That's why it can be in all different types of the body. That's why cancer can happen to any cell. It's just one of your own cells that got out of control, right? And then it keeps going and going, and a tumor is just a mass of cells. And we'll talk about why a mass of cells can be so dangerous. And so here's, yay, all this stuff that's going to cure everything, and we need more money. See, all this good stuff, yay.
Right, okay, people.